Well, we continue our discussion of transcription, and I put this little image up here just to remind myself that there's some very nice YouTube videos and other videos on the web that you can look at that work through the process of showing how all of the pre-initiation, initiation occur, the recognition, and then the actual elongation and form. So I think you might want to take a chance to look through those. So we're working our way from genotype to phenotype, and we're at the first level talking about transcription. We looked at recognition, this idea that you have to go through this whole process of finding TATA, bringing in the TATA binding factors, those bring in other transcription factors and build a complex that in itself then attracts this secondary complex, which is RNA polymerase. So we're talked about how that works and recognition in a general way. So we're at the next stage, which is initiation, meaning that we've recognized the site, and we're going to pull in RNA polymerase and actually start the process of moving along the template strand to start producing a message. Okay, so we're at initiation. So initiation is binding of this RNA complex, sorry, I can't talk. And there's a number of transcription factors that come into play. You probably see in your text the transcription factors 1B and F and all these others, and RNA polymerase itself, the actual enzyme itself, as well as the complex that makes that up. It's all recruited in, okay? I'm not worried too much. Please don't try to memorize these for this course because it depends on whether you're in prokaryotic systems or eukaryotic systems or whether you're in early prokaryotic systems or mammalian systems. The numbers and names of these change fairly dramatically. The point is, you have to bring in these other elements though, okay? So this, for us right now, this is RNA polymerase. That's what this enzyme is. We've got the this complex, this recognition complex in place. All right, now you notice it says be careful here, all right? This strand that we're going to work with, we're going to call the template strand, and we're going to build RNA in a 5' to 3' direction, which means we read 3 to 5. So in this case, the bottom strand here is going to be the template strand, or the strand that we're going to actually read from, okay? In other drawings, it's sometimes the top. You really got to look to see whether or not you're 5', 3'. The other thing to be really careful about is this wording right here. It's just all over the place, depending on what textbook you're looking at, depending on where. Right? Sometimes they call it the sense strand, meaning template, the anti-sense strand up here, the coding strand. I mean, it seems like to me the coding strand, you know, we're going to try to stick with template strand because that's consistent with our textbooks and things. Template strand is what we're going to read off of, and coding strand or anti-sense strand is what the other side. Now, what's interesting is if you want to know what the message is going to look like, you just read the coding strand, the opposite strand, and replace the T's with U's, and you've got the message, right? But what we're actually going to read from, and RNA polymerase is going to use to build the new message, is actually this 3', 5' side over here. Okay, so just be careful with that. All right, now not only are we bringing in all of these things, we're also bringing in this what's known as a mediator complex, which is up to 20 polypeptides, forms, protein-like structures, okay, that are brought in. Included some serious proteins, which are ATPase, which is helping to obviously activate enzymes in its ATP form, as well as helicase. You may remember helicase back from replication. It's the same type of thing, right? We have to open up this DNA and form a structure. And that's exactly what happens when these come in, this mediator complex comes in. I showed you on the last lecture that really once you have started this process of recognition, the first thing that happens in initiation is you start to open up this double-stranded DNA, right? And when that does, it's called a transcriptional bubble. And it basically just unwinds about one round, okay? So we're going about 10 or 12 bases, 15 bases long, opens up. And that means we have to knock off all the nucleotides and pull those apart. And you have to knock off all the nucleosomes themselves and break those up and make the whole structure opened up, okay? Well, then you have the template strand. Now it's open and it's available. In this particular picture here that you're seeing, it's on the bottom, right? So we're going to read from across the bottom, and some of our later ones will read across the top. Doesn't matter. Just look for the 3' end of the DNA, and that's where we're starting from. All right. Now this gets very complex. It's not just this is going on. What's happening here is, remember those things called enhancers that we talked about as being way upstream of these genes? Well, the DNA goes off over here, way up somewhere. There'll be these other recognition areas that have to, the DNA will actually loop back and sort of form some complexes that help lock all this down. This is one model of what that looks like. So here's our TATA box. Here's our TATA binding proteins and all the complexes going on there. We've recruited in RNA polymerase and we started down. Well, RNA polymerase doesn't like to be on DNA. It falls off, it connects in. So these sort of acts as a cap, a large cap that kind of holds that RNA polymerase down there on that system. And it's linked back literally to some other areas, these enhancer regions and other things where you have other complexes going on. So just imagine that we're talking about probably 50 different proteins, certainly polypeptides, that are all active here just to get the transcriptional process going. And each one of these, of course, had to be coded somewhere in the genome and their products made message. And then if they are proteins or polypeptides, then they had to go through the whole process of translation, brought back into the nuclear region and all that. So this is a fairly, very complex process that's going on here. That's why I said you could spend a whole semester just studying transcription, and certainly transcription translation easily could take up a semester or more. Well, anyway, we have this complex. We now have RNA polymerase down here starting to work, and it moves down from the TATA box, and it will move a ways down before it starts to actually building the first bases in. But it builds, of course, it reads in a 3', builds in a 5'. So we're going to use the template. We're going to start to move along this direction. And this is, of course, the non-template down here. Okay? And the RNA polymerase will come in. It will try to move down anywhere from 30 to 50, 100 bases down before it starts. And then it'll hit the start of the transcriptional part and it'll just start transcribing, okay? And to do that, what it does is it brings in the first complex, brings in the first nucleotide that's complementary to whatever's on the strand up here. Then it starts to bring in the second one, and then it forms phosphodiester bond, and then it moves on. Now, in reality, it turns out that it may take several tries for this to happen. Sometimes the RNA polymerase system gets on there and then falls off almost, disassociates. And the process has to try again. That's why that capping is so important here. But initiation is bringing in an RNA polymerase complex, moving it down past TATA, getting it locked on, and getting the first couple of bases brought in. That would be what we would define as initiation. Now after that, we're just going to go from one base as the other, just like you would say replication, and just build phosphodiester bonds complementary to each one of these bases that you have on the template strand. Again, remembering that everywhere there's a T supposed to be put in, so an A up here, a T here, you put a uracil in place. So this is the process of elongation. We're simply going to move along and move the process like this. RNA pol is going to move down through here. It's going to read the strand, and it's going to produce a message. This end of the message will already start to be modified, and we'll talk about that modification in the next series of lectures. But we're actually going to modify this end of eventually modify both ends, but this one's available to start that process. Okay. So we're going to read along. We're just going to make nice little complements, and we're going to move, as we said, reading 3' to 5', but making a transcript that starts out as 5' first, and eventually ending up. And you need to think about that. Why is that direction important? Why would it go that way? Think about what 5' and 3' mean and how you link up those phosphodiester bonds, right? You bring in this triphosphated nucleotide, and it's using those extra 2 phosphates to help catalyze that bridge, and it does it by catalyzing because the 3 phosphates are hooked onto the 5' carbon, and you want to hook to the 3' up here. You can use that energy to do that. So that's why you build in this direction, just like you would for other things. Okay. Again, it's an exact copy, just reiterating what we said a moment ago, except that you're using thymines are being replaced with uracil. And of course, the nucleotides themselves are made up of ribose, right? 5 carbon sugar, same, but it's got that extra OH on the number 2 carbon. So think about what you're producing here and how you might get it. All right, to kind of go back to our model of what it looks like, RNA pol simply moves down through the system. You've got a growing strand. It looks something like this, and you've got this message being made, single-stranded message being produced. In many textbooks, your book kind of shows it like this. Okay, moving along. Now remember, there's lots of things that have to be happening here. You've got to be opening up the DNA. You've got to be removing all of these nucleosomes and getting that released. So you've got to be moving down through here, and hat's got to be acetylating these structures and causing them to break apart. And then as soon as you go past it, you start to build them back, unless you're just going to read that gene again. Now, sometimes you do. Remember, we've got these cat boxes and other elements that kind of regulate how many times you go through this process. Well, if you're going to go through multiple times, you're going to make multiple messages, and it'll stay uncomplexed as sort of naked double-stranded DNA. It'll form back double-stranded immediately, but it'll stay without the nucleosomes. If not, then you've got to form all these nucleosomes back, right? Because you've got to wrap it around. So behind all this is a whole complex of proteins that are coming along, rebuilding this whole structure as we move further and further down. So we would read through here, and most genes, as we said, are minimally 1,000, 1,200 base pairs long. So we read that up to 15,000, 5,000, 15,000 or more, up to sometimes 100,000 bases. But however long it is, you start to read. So the question is, how do we know to stop, right? How do we know where we're at the end of all this? We're just reading DNA. What does it know? Well, that's the process of termination. And termination is cleavage of the transcript, but it's also, before you cleave it, you have to recognize where you're supposed to be terminating, right? So that's part of the process. And what we're looking at is, remember when we talked about that poly-A signal, right? This is obviously a poly-A signal in the message itself. There's no uracil in here, but we're really looking at a T, and we're going to have, right here, we're going to have a T there, right? So we're going to have T, all that through there. Recognition of that in the DNA itself. And what that does, that signal, it's a motif that we're going to use later when we process the message itself. But in this case, what it does is it brings in a couple of very important other products that are going to start to initiate the process of removing all of this complex. Okay? And there's going to be a release and a cleavage stimulation factor that are going to play a role. And actually, sometimes what happens is you read past this and then you just cleave off the actual message itself. And there's all sorts of things that can occur depending on the process, okay? And then there's this release factors. You basically are going to break this whole thing apart, okay? That's what we're trying to do with these release factors. So it looks something like this, kind of a model of it. Here's our system with our T's and A's and all of that. Very often the end is also GC rich. And when we start looking at these genes, we'll see that for many of them, they're actually GC rich. And so what will happen is that it'll form a hairpin. The single-stranded DNA will actually bow out and cross bond between the GCs, and that basically stops the whole process. But as we said, in coming into all this are these release factors, which are these combination of polypeptides, proteins that basically disassociate everything. And then, of course, on this end, right here, back up stream, you start forming your nucleotides back up. You start to bring in these, the nucleotides have fused back together, and you start to bring in all of the histones that you need to reform all of these beads on a string structure that you would have. Okay? All right, so that's basically the process as far as we're concerned. Very cartooned. Again, when I just say factors, that's a generic term we're throwing out here. There's very specific names, very specific things that go on, but it really depends on the gene. It depends on the organism, the level, whether you're talking about is happening in yeast or whether you're talking about happening in C. elegans or fruit flies. They'll be different. So that's why we're not spending a huge amount of time worried about it here. We're trying to just get the process down, right? So what have we talked about? We talked about recognition, right? Which is recognizing the TATA, bringing in all the elements that are needed to TATA binding protein and all the complex there. Once that's established and you've got that, you initiate by bringing in RNA pol, locking that RNA pol down with a cap. Then you start, RNA pol starts to move away. It's downstream from TATA anyway, moves down a few bases, and then initially starts to build your message, first bases of your message. Once it gets the first few bases on, you just start the process of elongation, which is movement around. Eventually, you hit these poly-A recognition sites, and that brings in release factors, which cleave off the message itself, and bringing up, so now you'll have sort of a naked 5' to 3' single-stranded message, and all the DNA will start to form back into its double-stranded components. Right? So at this point, what we've done, we've taken DNA, we've transcribed it, and we've made message. Now, right now, we're talking primarily about what's called mRNA, messenger RNA, right? Messenger RNA is what carries the genetic code for our purposes. Okay. And at this point, and this is important, we're going to call it heterogeneous mRNA. Okay, heterogeneous mRNA. Why? Because it's still at this point, transcription is just a blind process. Once it actually starts, once you initiate it, it reads every base that's in that transcriptional unit. It doesn't know whether it's or introns or exons. It doesn't care. So the message at this point, when it's first produced, is full of introns and exon coding that you have to splice out and remove. So we call it heterogeneous RNA at that point. We're going to modify it. Okay. Other things, though, are transcribed. Remember, we said there's the classical gene, which is meaning that piece of DNA that forms these messages that'll make protein. But we've got to make a ribosomal RNA, has to be transcribed, and it turns out it gets modified. tRNAs, which we're going to see in a moment, are going to be used in a moment being next series of lectures when we talk about translational process. They're going to be used. They're actually modified as well. Then we have all sorts of other RNAs that get read off, and these are transcribed. So you could think about the units that they come from as being, at least at the molecular level, genes, and they're important. They're microRNAs. There's all sorts of things. There's small interfering RNAs, which is a really interesting one we'll talk about later. You get all these messages down into the nucleus, out of the nucleus into the cytoplasm. You just can't have those messages pile up forever. You've got to do something with them, right? So you start to break them apart and recycle the nucleotides. And one of the ways that happens is through these small interfering RNAs. They trigger a degradation of those messages so that you don't just pile them up forever inside of the cytoplasm. That's an interesting thing we'll talk about later. And we'll talk about some of these nucleolar RNAs and other things as well. All right, for us, that covers transcription. I would suggest you go read about it. Well, certainly you'll watch a few videos on it, get a good handle on what's going on. But for us, that covers enough about what's going on. We've taken our DNA, this very important piece of storage information, a huge amount of information we store, and we've read some information off of it that we can now use to work our way down toward finally producing protein, finally making expression of what's in that code. And before we can do that, though, we've got to take this heterogeneous RNA and convert it over to what's known as mRNA, which very often is used as a mnemonic to say, instead of messenger RNA, is to say matured RNA, right? So we're going to mature the message and get it ready to be moved out of the nucleus and brought out to where it can actually be used in the transcriptional process.